A very good day to all the assessor. My name is Win Win and my topic today is exosome-based vaccine, their position in vaccine development against infectious diseases. This is my content. What are exosomes as an introduction and the roles of exosomes in infectious diseases, exosomes in vaccine development, followed by exosome-based vaccine studies for various infections, the challenges and conclusions. Exosomes are nano-sized cell-derived extracellular vesicle packets of information which carry bioactive cargoes such as proteins, microRNA, RNA, DNA interleukins, and lipids. This lipid membrane structure, cargo's carrier, comes from different cell types and possess crucial function as mediators for cell-to-cell communication and involved in immunoregulation processes due to the ability to transport various molecules to near or far targets to exert different biological functions. Extracellular vesicle is actually the big family where exosome is one of it. There are mainly three subtypes, which also you can see in this picture is either exosome with a smaller size of 30 to 115 nanometer, whereby the other two is bigger in size. Okay, they are all defined as a lipid vesicle secreted by cells. So what is the main differences of exosomes from the other two types is that the other two types are just shedding from the plasma membrane, whereby exosomes originate from the endosomal membranes located inside the cells. Now let's go to the biogenesis of exosome. Okay, so you can see from these two pictures how exosome comes about from the intracellular part. We started from the intracellular molecules swapping with early endosome materials, and they were recycled to formulate endosome. After that, followed by formation of multivesicular bodies, and lastly they continue to sort cargoes with formations of intraluminal vesicles, and the intraluminal vesicles will then be released into the extracellular space, which is also known as the exosome. And they are released through the endosomal, mainly through the endosomal complex pathway, which can classify into ESCRT 0, 1, 2, 3, and also if it's non-endosomal pathway known as the AAA ATPase VPS4 associated complex. Exosome can be found in all kinds of bodily fluids in humans. As from this picture, you can see it can be found in saliva, blood, urine, milk, and so on. There are two methods of how exosomes deliver their cargoes to their recipient cells. First one being direct fusion with plasma membrane of the target cells and deliver the content into the cytosol. Second one, attachment with molecules on the surface membrane of the recipient cells, such as major histocompatibility complex. The protein content is a major component in exosome, whereby can be classified into specific and non-specific. The non-specific proteins are cytoplasmic enzyme, heat shock proteins, cytosolic and transferrin proteins. When it means by non-specific means that all kinds of exosome does have all these proteins, whereby there are other specific proteins such as signaling proteins, antigen presenting proteins, immunoglobulin, cell surface peptidases, and tumor antigens. The specific proteins in exosomes are the one act as the markers to determine which parent cells that the exosomes are coming from. There is also an abundance of cholesterol and phospholipids in exosome, which have a function to maintain immune homeostasis. There are also different types of RNAs inside exosomes, such as the mRNA, microRNA, long non-coding RNA, which they will be delivered to the target cells, and this process is involved in gene expression regulations. Exosomes are, besides human, are actually found to be secreted by almost all living creatures, such as viruses, bacteria, fungus, and parasites. The pathogen actually use exosomes to help them replicate, transmit, and also virulence coordinations. There are two types of exosomes in pathogen, one which exert communications between the pathogens and the other one between pathogens and host cell. There is a study shows that there is an increase of exosome productions and changes in their cargo contents during an infection in vitro and in vivo. The exosome in the pathogen acts as a two-edged sword. Why we say so? Because first, it aids the pathogens in spreading and invades the human host cell. On the other extreme, it actually downregulates the anti-pathogens mechanism by mounting immune responses. Exosomes are recognized by immune cells due to the surface protein on the exosomes that are present, which includes T and B cells receptors, cytokines, integrins, and cytokines receptors. Exosomes are derived from CD4 and CD8 can actually strengthen the functions of B cells. Exosome from antigen-presenting cells such as macrophages with MHC1 and 2 actually promote T cells activations and proliferations. Exosomes from the N1 type macrophages can polarize macrophages to N1 type, inducing cellular and humoral immune. Exosomes from mast cells also transport functional RNA and other immunomodulatory proteins, and this helps the maturation of dendritic cells and proliferate T and B cells. Exosomes from dendritic cells and B cells with antigenic peptides also induce adaptive immune responses. So you can see that exosomes are the immunomodulation properties of exosomes. Therefore, why exosome can be a good candidate for vaccine development, both as therapeutic and preventive, due to the properties of the antigen delivery and immunomodulations. Besides, exosomes also contain heat shock proteins, which function as antigen presentation, attaching antigen peptides onto the MHC, help with dendritic cell maturation, and also involved in the NF-kappa B translocation into the nucleus via CD91. And exosomes are highly biocompatible due to its natural endogenous carrier, and also no doubt high biosafety and cause low immunogenicity and don't provoke any harmful immunological reactions. Exosomes are also safer than virus-based vectors as they don't replicate, but like viruses, they can selectively absorb cells via surface factors, which help increase therapeutic effect in target tissue with minimal system side effects. Due to the advancement of technologies, there are many ways to enhance the exosome-based vaccine efficacy. Here are a few listed. Exosome can be implanted with selective antigens done ex vivo or use engineered methods to alter the composition of the exosome. Another one being manipulating the antigen presenting cell through pharmacological methods or genetic techniques, or can also incorporate adjuvants or cytokines which has immunostimulatory functions. Another one being the vaccine can also be amended with microRNA or dendritic cell targeted molecules to enhance this specificity. Now let's go to the studies done on exosome-based vaccine for parasitic infection. Malaria parasites are a devastating infection which invade cells like red blood cells, liver cells, spinal cord, gut endothelium, and saliva glands. There is a study shows that found that there's a high number of circulating exosomes derived from Plasmodium falciparum and Plasmodium vivax isolated from patients. The exosome from the pathogen can actually fuse with endothelium and monocytes via endocytosis, and this changes the vascular properties and enhances the DNA sensing pathway via microRNA to access its virulence factors in human hosts. There is already studies shows a successful exosome derived from malaria-infected red blood cells managed to immunize mice against the deadly Plasmodium yoli in year 2011. These vaccines comprise of HLA1 and parasite antigen able to activate non-exhaustive memory T cells. There are ongoing efforts in aim to develop an effective and lifelong anti-malarial vaccines, along with considerable interest in improving vaccines delivery using exosomes and new technologies for mass productions of exosomes, such as the exosome mimetic nanovesicle. Toxoplasmosis is another severe infection which can cause fetal complication in pregnant mothers contracted with primary infection. There's a study shown that infection before pregnancy can actually help fetus build immunity against it, therefore suggesting that vaccination before pregnancy might block vertical transmission of the parasites. Exosomes derived from dendritic cell, short for known as DEX vaccine, from the spleen containing the Toxoplasma gondii antigen in mice injected before pregnancy, shown to offer protection in pups as there's fewer cysts found in pups' brain. Besides, there's also a robust humoral responses in serum in vivo detected with cytokine production by splenocyte during proliferations. Leishmaniasis caused by the parasite Leishmania can secrete exosomes to modulate innate and adaptive immune responses through macrophages and dendritic cells. So exosomes derived from dendritic cells loaded with antigen can induce protective immune response against the Leishmania major after coupled with paraformaldehyde or exposed to UV radiation in vitro. And this study shows that this vaccine lead to T cell proliferations and interleukin-12 secretion by splenocytes. Besides, the macrophages with Leishmania exosomes were also found to induce the secretion of interleukin-8, and this has suggested a broad-based mechanism of protein export by Leishmania. Hence, the development of cell-free vaccine for prevention against Leishmaniasis is workable. Let's go now to bacterial infections. MRSA is a methicillin-resistant type of this Staphylococcus aureus. Is still under high priority watch list of antimicrobial resistance bacteria, most threatening to humankind under the WHO in year 2024. So, there is studies show that the staphylococcal alpha type phenol soluble modulins can induce release of exosome by interrupting the host cytoplasmic membrane. Autolysin activity and peptidoglycan cross-linking also promote exosome production by changing the host cell wall permeability. So, genetically engineered exosome from Staph aureus mutant actually successfully express the detoxified cytolysins, which trigger immunity in mice through cytolysin neutralizing antibodies, protecting those animals from deadly sepsis models. These again highlight the usefulness of exosome as a vaccine platform against Staph aureus. Another deadly bacterial infection known as tuberculosis, caused by the pathogen Mycobacterium tuberculosis. There is positive result of exosome derived from Mycobacterium bovis and Mycobacterium tuberculosis. Infected macrophages containing antigen lead to an activation of both CD4 and CD8 splenic T cells in mice models. Besides, there are also memory CD4 and CD8 T cells detected following the intranasal administration of this exosome vaccine in mice. What about exosome-based vaccine for viral infection? There are attempts of the first HIV vaccine using exosome deriving from dendritic cell expressing HIV-specific glycoprotein GP120 was called GP120 Texo. This vaccine shown to activate robust and long-term HIV-specific CD8 T-cell immune protection in mice. Soon after, another vaccine was designed to stimulate immune responses against GAP protein, which is in charge of the HIV maturation, and this vaccine is known as GAG-Texo. It's tested in animal models with chronic HIV infection and shows to induce GAG-specific immune responses, suggesting cytotoxic T lymphocyte activation to attack HIV-infected cells. Hepatitis B virus is also another fatal infection causing complications such as liver cirrhosis and hepatocellular carcinoma. The various exosomal microRNA from hepatitis B virus involvement in viral host interaction through regulating gene expression in host cell, and this gave a useful insight into understanding HBV pathogenesis pathway in hope to develop a novel therapeutic approach against this virus, including vaccine development. In addition, there are ongoing studies in early phase investigating exosome as adjuvant coupled with recombinant HBV antigens, and these studies reveal positive results whereby this vaccine formulation is capable of stimulating T helper 1 immune response and thus increase the interferon gamma levels in mice. Now let's go to exosome-based vaccine for fungal infection. Fungal exosomes are actually shown to be an ideal vaccine candidate due to the nanometric size, and is also a natural transporter of various antigens and coupled with sugars, and this help in cell recognition. Candida albicans in the human digestive system has dual role whereby it acts as a commensal fungus or opportunistic pathogen. The overuse of antibiotics leading to a disruption of gut flora causes an overgrowth of these Candida albicans. Entering into bloodstream can cause severe diseases. There are also studies of exosomes from different types of Candida species, consist of different types of immunogenic substances, which indicate that exosome can be used as potential immune regulators to fight against this fungus. Candida humuloni species is also a multi-drug resistant strain pathogen, can cause superficial and invasive infection in vulnerable populations. Low concentration of exosomes derived from the pathogen are shown to escape the recognition of the oxidative burst pathway by macrophages, and hence exosome carrying the antigen proteins can be exploited as potential vaccine candidate against Candida species. Cryptococcosis caused by Cryptococcus, also a common opportunistic fungal leading to severe disease when affecting the lung and central nervous system. Web protein from exosomes of Cryptococcus neoformans in mouse have shown to activate adaptive immune response without even adding a carrier or adjuvant. Cryptococcus neoformans with CAP59 SG11 mutation in another study also possess properties suitable for attenuated vaccine development. All the previous studies mentioned shown that exosome-based vaccine it's a very potential anti-infective agent, as it shows good immunogenic responses and thus help prevent infection throughout the microorganism, including bacteria, fungus, and parasites. Due to the unique properties of exosome in immunomodulations. The current challenges of exosome-based vaccines is that all the research studies, as at to date, is still at primary stages. There are still more to study and explore to advance this type of vaccine into clinical stage. Different techniques are still required to be explored to couple with specific antigens or antimicrobial molecules to exosome-based vaccines. There are also gaps in standardization protocols on isolation and synthesis methods of exosome. More studies are still required to understand the pharmacokinetic of exosome-based platform. Innovations thereof are required to design high-efficacy vaccine and in order to develop robust and yet antigen-specific immune responses. There are also still lack of knowledge of large-scale immunization programs, and also extensive applications yet in clinical settings. It is now well evident that cells infected by external pathogens can release an exosomes, and exosomes also have many involvement throughout the process of infection. The dual roles of infectious biology of exosomes, including spreading or limiting an infection, depends on the different type of pathogen and its target cells. Again, an exosomes are shown to be a potential candidate for future vaccine development in both protective and therapeutic therapy, and the field of this research is dynamic and vast with ongoing changes according to various demands. However, and quite positively, that exosomes will be in near future enhance the efficiency of vaccines, and is a promising platform to invent more novel vaccines for human race. These are my references. Thank you for your kind attention.